Good morning or good afternoon, wherever you are. It's a beautiful day in Charlotte, North Carolina, where I'm sitting. My name is Roger Giles. I'll be your host for the next 45 minutes. The first webinar of 2015 for Ground Bio 1. We've got a series lined up for the rest of the year, but we're delighted to be able to offer you this webinar, which is entitled A Different Approach to Achieve Higher Transfection Efficiency. So before we start, I'd just like to ask you to check your speakers, make sure you've got the right volume setting on your computer, and if you do have any problems with the sound, please write a note to me on the chat box, and I'll best to help you out. We've presenters today, good friends of mine. The first is Géraldine Guérin-Peyrou from Polyplus-transfection, and also got Dr. Thomas Schreiber from Bio 1 in Germany. Obtained her biotechnology engineering degree from the renowned French school ESBS, the MSc in biology at University of New York in the US. She gained expertise in the fields of cell biology and genetics by spending 4 years in researching the role of histone in mice or mouse models of hereditary brain cancer. Ms. Rowe's work has been published in molecular cell biology and entered at several international meetings. For the years, she has led Polyplus-transfection's global team of technical support specialists. She manages the development of new, innovative transfection products. Dr. works for Ground Bio 1. He studied chemistry at the University of in Germany and at the University of Toronto, Canada. PhD at Tübingen University Medical School, knowledge in cell biology and actions of stem cells with their microenvironment. As a fellow at the National Medical Science Institute at Riedlingen, Germany, he found applied R&D in the fields of genomics and bioanalytics. At one, Dr. Schreiber is responsible for the development of intensive cell culture. Now over to Géraldine, who will continue her presentation.
Good afternoon, everyone. Thank you, Roger, for your kind introduction. So today, I'm going to introduce you to the basics and principles of transfection. We will then present experimental results on how to improve transfection efficiency. A discussion about how place can improve transfection efficiency, and I will conclude our presentation with a short troubleshooting and Q&A session. If you have any questions or you would like to be answered, please feel free to send a chat to our host, Roger, and we'll answer all of them at the end of the presentation. Transfection is the introduction of material, mostly nucleic acid, into cells. Usually people will transfect on cells, but insect cells can also be transfected. Cells of amoeba, fungi, or yeast cannot be transfected. The thing about transfection is very important to remember that nucleic acids cannot enter cells by themselves, and I'll show you in a few slides why and how to overcome this issue. Nucleic acids can be transfected from the very common standard DNA or small interfering RNA all the way to more molecular molecules such as microRNA or messenger RNA. So why do people perform transfection? Usually to overexpress a gene of interest to be able to analyze its function in specific type or to hit a specific gene or a specific regulation system. But more and more, actually, scientists use transfection or transient transfection mostly for production of large-scale protein and viruses, industry purposes, all the way to industry purposes. There are some major transfection methods. The first one is really physical. It's putting nucleic acid into a cell through a hole. So we made by micro-injection of DNA, for example, directly into the nucleus of cells. You can use electroporation that locally stabilize the cell membrane through electric field, creating a small hole into the cell membrane, allowing nucleic acid from the external medium to penetrate into the cell. Techniques have drawbacks, and so there are ways of getting nucleic acid in cells by floating natural mechanism involved in endocytosis. So one natural mechanism to get nucleic acid in cells is viral infection. It's well known that viruses can inject the genome into a cell through the infection process. It's a very efficient way of getting DNA into a cell. It's highly specific of type of cell, depending on the virus you use. It has some drawback. It's quite cumbersome. You need to produce the viruses that request steps, as well as risk of immune response. So back in the 70s, scientists have started working on developing molecules that could be used as a vector for transfection. Two large families of molecules have been used, lipids or polymer-based transfection reagents. The idea here was to use molecules that can mimic the advantages of the virus without being infectious. I'm really going to focus on chemically mediated transfection, as this is really our core business as part of transfection. Our company is specialized in transfection reagents, and we have developed a wide spectrum of different transfection reagents depending on the application, as well as cell targeting and the type of nucleic acid transfected. For this, we have a wide range of different transfection reagents, from our Jet Prime, which is used for gene expression experiments, all the way to high throughput screening application or bioproduction with our latest vector flow. But I'm really only going to talk about gene expression, and I'm going to walk with you through the different steps that are fundamental when talking about transfection. So, just to the beginning of the story, why do people have started on transfection reagents, and why do we need transfection reagents? Well, acids such as DNA or siRNA are negatively charged molecules, and the cell membrane is also negatively charged and hydrophobic. So there's a way that the nucleic acids can interact through the cell membrane. There's a relation between those two entities. So the transfection reagent is that by adding a next step of positively charged molecule, you could form transfection complexes that will be turned positively charged and hence interact through ionic interaction with the heparan sulfate from the cell membrane and then leading to transfection. That's really why people have started developing transfection reagent back in the 70s. But what I want to walk you through today are the different steps of transfection. First, to form those transfection complexes. Then how the complexes can enter into the cell. Then how the transfection complexes will release nucleic acid into the cytosol, and how this nucleic acid can reach the nucleus for expression in the case of DNA. So visualization of those cationic complexes. An easy way to visualize nucleic acids and transfection complexes is actually to look at them through electron microscopy. And this is indeed what it is. As you can see here, you can see a long string of DNA. And as you add a transfection reagent such as Jet prime, you form those cationic complexes that are Dutch shaped. So it's easy if you have an electron microscope on hand, but there are easier and cheaper way actually to visualize transfection complexes. Them is very easy. It's to use an aqueous gel cleaned with acidium bromide. So here on this gel, what you can see is a constant DNA amount, a complex with volume of jet prime transfection reagent and loaded on the gel. When you use a little amount of transfection reagent, the nucleic acid is not complex, is under the form of this long string, and then it runs into the gel, and you can see the nucleic acid at the bottom of the gel. When you have a sufficient volume of jet prime transfection region, you form the transfection complex, and it's actually stuck in the pocket of the well. So it's also a very easy test to perform if you're using a new medium or a new transfection reagent or a new component in your experiment, and you want to make sure that you actually complex efficiently with the nucleic acid handling for efficient transfection. So cell form now is to enter into the cell. Here again, I'm showing you a picture of fibroblasts. They were transfected with polymer-based transfection reagents. Here you can look at the different steps of cell entry. You can see the transfection complexes that are binding to the heparan sulfate. Then there will be the significance from the cell membrane will interact. The PK-mediated phosphorylation, heparan sulfate will lead to the actin binding to the protein and the creation of those crypts, allowing for the engulfment of the transfection complexes into the cell of the cell. And that transfection complexes are inside the cells now. In which the acid has to escape from the endosome. And there are ways of doing this. Usually the escape from the endosome is different if you're using a lipid transfection reagent or a polymer-based transfection reagent. If looking at the mechanism of lipid-based mediated transfection, you can see that the DNA is stretched between layers of lipids. This lipid can then interact with the lipid from the cell membrane to form an equilibrium that will be created between the lipids from the transfection region and the lipids from the cell membrane. The equilibrium will allow for fusion of two lipid bilayer. Tunnel in the cell membrane, allowing for the nucleic acid to penetrate into the cell. The main drawback of this technique is you actually create locally holes in the cell membrane. So those nucleic acid to enter, but also the component from the medium that side of the cell. And the reason why a lot of lipid-based transfection reagents from generation would not be efficient in the presence of serum and antibiotics. Those particles could penetrate into the cells at the same time as the DNA. Looking at polymer-based transfection is actually our specialty at Polyplus-transfection. The polymer will associate with the nucleic acid to form the positively charged complexes that will then diffuse and sediment on cell surface, where they can bind to the heparan sulfate and taken by endocytosis. Once in the endosome, polymer that Polyplus work with has the ability to burn the pH inside the endosome, allowing for gentle release of the nucleic acid into the cytosol via osmotic lysis of the endosome. It's really gentle way allows polymer transfection reagents to be a lot more gentle in cells than lipid-based transfection reagents. And indeed, this is something we saw when performing tests on the advanced PC plate from Géraldine Guérin-Peyrou. We heard transfection efficiency and solubility following lipid-based transfection and polymer-based transfection of immortalized fibroblast IMR90. And you can see on this picture that the cells are a lot healthier on following jet prime mediated transfection than lipid-based mediated transfection. So main cell characteristics for an efficient transfection. I want to work with fast-growing cells because usually the gene is released into the cytosol of the cell, and it will reach the and the cell divide. This will occur through cell pore, but only during mitosis. Hence, it's very crucial to work with cells that divide actively, not accretion cells such as primary neurons, for example. Second point is that adherent cells are always easier to transfect than stem cells because they express more heparan sulfate on the cell surface, allowing better arrangement of the transfection complexes to the membrane of the cell and higher efficiency endocytosis. And is the physiologic state of the cells themselves. It remains a traumatic event for your cells. You are going to push nucleic acids into them and have it overexpressed. It's a natural mechanism. And primary cells are usually a lot more sensitive to transfection than cell lines could be. When transfection, there are different criteria that are crucial for researchers. So in a high transfection efficiency, but also high protein expression that will be homogeneous between the different cells. TB and significant error are critical points if you like to publish in high-impact journals. And of course, low toxicity will guarantee you results that you can trust. And the results are due to cell death. With all parameters in mind, I wanted to show you the protocol for transfection. So, plasmid DNA is diluted in jet prime buffer, and jet transfection reagent is added. The mix is mixed, and then just added to the cells. It's a very straightforward and easy protocol, but there are actually a few steps on which you can act to make sure that your transfections will be more efficient. And of course, the amount of DNA, the volume reagent that you use, the two complexes, and of course, the ratio used between DNA and nucleic acid and transfection reagent. But also, very important is all the logic state of cells. The way you have operated their cell density, the increase time, and the volume of medium are critical parameters. But we would like to tell you more about is how a good plate coating can make a difference on your transfection efficiency. And with this, I'll let Thomas Schreiber from Guérin-Peyrou introduce the results we generated together.
Thank you, Dean. So with this slide, I'd like to make you very familiar with a couple of cell culture surfaces. Besides the standard surfaces that are used for adherent cell culture or suspension cell culture, Guérin also features what we call advanced cell culture surfaces that are non-biological and animal-free. These are called advanced TC or cell repellent. Advanced TC is the surface adherent cells, and cell repellent cells can be used to create spheroids, for example. Furthermore, there are several biological surface coatings, the brand name Cellcoat, which are, for example, polylysines, collagen type 1, fibronectin, lamin, or double coatings. Today I'd like to present you some results on the surfaces that we use, which are standard surface, advanced TC, and polylysin. To investigate the transfection efficiency with different plate coatings, we started with the transfection of a GFP vector into HeLa cells. Experiments were performed in 24 well plates, and 1 per liter of jet prime transfection reagent, as well as 0.5 micrograms of GFP vector DNA were used per well to transfect the HeLa cells. The bright field microscope pictures on the left of each group and the fluorescent microscope image on the right. Even for this standard HeLa cell line, the effect of the cell culture surfaces is easily visible. Cells cultured on polylysin or advanced TC put it better, as you can see with the number of cells, and also since there were more cells, they also expressed more GFP. The transfection efficiency per given number of cells was then assessed by flow cytometry analysis of GFP positive cells, this shown here, and that was by 10 to 15% higher for the cells cultured on polylysin or advanced TC compared to the standard CELLSTAR surface. From this transfection efficiency, we also wanted to see whether the protein activity would be affected by the cell culture. Cells were transfected with jet prime again. Luciferase reporter vector was used in combination with the 24 well plate setup. After the transfected cells for a given time, the catalysis of chemical reaction with a substrate leads to the emission of light that was measured then in a plate reader. With the standard CHO cell line, and you can see that when entering the cells on advanced TC or polylysin, the amount of luciferase secreted or the of catalysis that place is higher in comparison to the standard TC surface. Results were obtained with the hepatocarcinoma HEP2 cell line. for human umbilical vein and the ciliary cells. The Luciferase units that were measured in the assay could be increased by culturing the cells on advanced TC in comparison to standard tissue culture. The Store cell line SHSY5I also gave a similar result, and the amount of Luciferase that was actually active was 3 times as high approximately with advanced and poly-D-lysine in comparison to the standard TC surface. Overall, we would see a better transfection efficiency as well as a higher activity of Luciferase when advanced TC or cell-coat poly-D-lysine plates were used. What could be an explanation for the observed effect? Polystyrene is usually used for cell culture disposables, and by itself, the polystyrene is very hydrophobic due to the absence of groups at its surface. So most cells would not attach or spread, and also proteins could not bind efficiently. From this situation, physical surface treatment is usually done, such as atmospheric corona or vacuum plasma treatment, and these are used for the activation of the surface by using then functional oxygen and nitrogen containing groups into the polymer backbone. In addition, the surface treatment leads then to the formation of an overall surface roughness, and in this results then increased hydrophilicity and also wettability of the cell culture surface. And this standard tissue culture treatment, we randomly introduce functional groups. GRANION also offers a more directed surface modification with advanced TC and polylysine. TC is a polymer modification that results in a net positive surface charge at logical pH, which is very beneficial for sensitive or stressed cell culture. Polylysine and poly-L-lysine have a similar effect, while these being biological molecules that are produced synthetically. They have to be applied as a coating to the surface, and this is done with minimal batch-to-batch variability as part of Greiner's cell coat manufacturing process. Let's have a more detailed look at the cell surface to understand more about cell-substrate interaction. Surround themselves with a complex extracellular matrix that consists of proteins such as collagen, elastin, or laminin, and these can interact with glycoproteins like fibronectin, for example, and receptors such as the integrins. Important for the initial cell attachment that is vital for transfection are the proteoglycans. Extracellular matrix proteins that have carbohydrate structures such as glycosaminoglycans attached to them. These other polysaccharides usually account for a negative charge that is in a biological setting very important for the integration of the extracellular matrix. For cell seeding and transfection scenario, this negative charge is important for the cell attachment. The step in the process of attachment is the interaction of the negatively charged cell surface with functional groups of the cell culture disposable. From this, it becomes pretty clear that the positive net charge of advanced TC or polylysine surfaces exert an electrostatic attraction to the negative cellular surrounding. Step would be if medium containing serum is used, that the serum proteins that are deposited on the polymer would engage with the integrins on the cell surface. And a third step would be the extracellular matrix proteins that are either applied on the cell culture vessels or are synthesized from the cell would then also come into interaction with the integrins on the cell in membrane. For it's important to know that the functional groups of the cell culture further interact with the cells for the adhesion process. So with the past few slides, to give you a better understanding of why the cell culture surface can be very important if it comes to tweaking your transfection, which means by choosing the right surface, primary and long-term adhesion of the cells can be optimized, and the cells have a more in vivo-like morphology than a better end, but goes along also with an accelerated proliferation, which means that the more cells are actually present for transfection at a given time. Reagents require the use of serum-free conditions, which usually requires the use of advanced TC or cell coat surface, because cells highly depend on the serum in the medium. As you can see in the combination with jet prime, a cytotoxicity could be achieved, for example, in comparison to lipid-based transfection agents. And overall, this then resulted in a higher transfection efficiency, what we obviously want to obtain. Last few slides, I just briefly address some troubleshooting issues when it comes to transfection. Well, one area is that the transfection efficiency is very low. So if you experience this, it's also advisable to check the transfection conditions. As Géraldine has already said, transfection can be quite traumatic for the cells, and therefore the amount of DNA applied should be as low as possible, as well as the amount of transfection reagent. So if you don't have a clue on what's going on in your system, then it might be advisable to try different combinations of DNA ratio versus transfection reagent to find actually the best conditions with regard to transfection efficiency, but also with regard to cell viability. Also very common is that cells are cultured for a very long time, and you probably know that cells own time usually alters the cells or the cellular behavior. So it's always important to keep an eye on the cell passage number of successful transfection. If cell viability is an issue, then of course the first two issues I just mentioned apply also here. In addition, the quality can be very crucial, which is basically should be a common standard in the cell culture lab to check for the serum quality. What you do is that you obtain several batches of serum and then try one fits best to your system and then stick to it for a long time. So serum issue, and as I mentioned already, the low amount of DNA and transfection reagent can be crucial if you have very sensitive cells. As I tried to show you, the plate surface can also be very important if it comes to viability. If you have sensitive cells, then you probably needed like a more sophisticated surface, such as advanced TC or polylamine, because the cells just happy on such a surface, for example. So with this, I'm actually at the end of my part, and yeah, Jean and me would actually be happy to answer your questions. And with this, I'd hand over to Roger again.
So much, both of you, for that very interesting presentation you've given us. During time, we have received several questions, and I would like to take this to go through that. Start with, there's a question here for you. It's to do with the jet prime. Do you have a list of cells successfully transfected with Jet Prime?
Indeed, we have. There's a list of cell lines that were transfected with Jet Prime. It's available in our cell transfection database on the Polyplus website. Over 150 cells and cell lines available there as references.
Thanks. And just on that point, obviously you can all read both the GranoBio1 and the Polyplus-transfection websites for any additional information. And this web obviously also being recorded, so that recording will be on both those websites going forward. Another question here for Géraldine. Transfect other types of nucleic acids with Jet Prime?
Indeed, Jet Prime has been specifically developed for DNA, but it can be used for siRNA, siRNA plasmid, mRNA plasmid, mRNA. It will be used for co-transfection of DNA and siRNA or DNA and mRNA. And it is perfect agent for CRISPR-Cas9 technology as well.
Dr. Question for you. Which cells have been successfully cultivated on advanced TC?
We got a pretty overview of cells that had been successfully cultured on advanced TC in one of our publications. It's called a forum article. It's kind of a review with regard to advanced TC. But to briefly summarize, we had cultured capillary cells such as neuronal cells, or also stem cells, murine stem cells, for example, and a whole range of cell lines on advanced TC. But the detailed list can be accessed in our publication that is accessible via the website.
Here, how to improve the detection efficiency when transfection 5 plasmids together or are together? Who would that?
Question, Roger, please?
Sorry, it's how to improve the transfection efficiency when transfection 5 plasmids are together.
It's 5 plasmids.
Yeah.
Okay. So you're transfecting several plasmids at a time. It's important to limit the amount of nucleic acid, the amount of to small recommended amounts in the protocol. And the ratio between each of the plasmids has to be adapted depending on the expression level of each protein required. So if you're expecting fast plasmid to put virus, for example, you want to express a lot of protein from the capsid, but very few from the genome. So you need to have more plasmid DNA for encoding for the capsid than plasmid DNA encoding from the genome. So the ratio has to be really optimized depending on the application. I hope that's the question.
If, obviously, if the attendees want to ask more questions, they're welcome to reach out to us through email or through on their web pages. So thank you for that, Géraldine. Dr. Question, what's the difference between advanced TC and D-lysine?
Well, advanced TC is a polymer modification, whereas poly-D-lysine is a coating. In terms of storage or shelf life, both feature a shelf life of 18 to 24 months at room temperature, so they are basically easy to go and do not distinguish a lot from standard cell culture ware.
Another one for Dr. Schreiber. My cell system runs on collagen. Substrate improve transfection as well?
Probably it would. We have similar data as the ones we presented today here that show that the beneficial effect of cell collagen type 1 is also seen with regard to transfection efficiency. Furthermore, we have also shown that as siRNA transfection efficiency increases as well with all the three strains, advanced TC, cell coat, or the cell coat collagen type 1. But yeah, you could really inquire for any further information on that.
Perfect. We are almost out of time, so what I'm going to do now is all the questions that were not handled today will respond via email to the actual question. And again, those answers will be displayed on our web pages. Just a couple of things. We have more information for you on our databases. So just refer to the web page. We've got FAQ databases. We have application protocols for both Grind 1 and Polyplus-transfection. If you would like to, just let us know. We can send you transfection news. We can also send you adverts for the next webinar series at Grounded Bio 1. So with that, to thank the presenters. I found it very interesting myself, and I'd also thank everyone that attended this webinar. If you'd like to help us, I really appreciate it if you could complete a simple one-page survey after you check from this webinar. We'd appreciate your feedback. And yeah, thank you very much and goodbye.